the first things that ecologists or demographers study when they're presented with a population that they're interested in is the population size. In our lab today, we are going to be using a method called the Mark recapture method. And this is a way to measure a population when you can't count every single individual. And for most populations, just doing a simple visual count is not going to be very easy. So we're going to look at a few different ways that ecologists might do this, might estimate their population size based on a smaller sample. If at all possible, it would be preferable to count the total number of individuals. Depending on the organism, this may or may not be feasible. So the larger the organism, the easier it's going to be to count. So taking a simple, straightforward visual count is going to be most useful if you have a really small area and if you have larger organisms, or some combination of the two. So for example, you would not probably try to count every single insect in a square mile. You might be able to calculate every single insect in a really small area. You may not. If you have larger animals, it might be easier to track them. It might be able to do some sort of video surveillance or drone surveillance or something. But in general, it's really hard to determine an overall count. And so usually what happens is that ecologists use sample size. They count all of the organisms that they can in a particular area to estimate the total sample size over a larger area. The other thing that they can do is sample a few individuals from a larger area and then extrapolate. So we'll talk about how they might do that. One of the ways that they do that is something called a quadrat or a transect. So these ideas are similar. They basically mark off an area that you are going to count all of the individuals that you come across in that area. So on this image on the side, you can't see the whole thing, but this researcher actually has a quadrat. It's basically a square. In this particular image, she has a frame. This one is just a PVC pipe frame. And what this is it's a standard area, and within that area, you count all of the organisms that you happen to come across. So what you would do here is you would lay out your frame over multiple positions, and what this does is just give you a smaller area in which to study. Now, depending on how big your area is, you probably wouldn't sample the entire area this way, but you may sample a small area and then expand that calculation out to the total area. The other option is a transect. So basically what this one is, if we have a forest and we have some trees in our forest, it doesn't have to be a tree, but maybe it's a tree in our forest, and you take a string and you count all of the organisms that you come across when you lay down this length of string. So you count every organism along this particular area. And if you are doing the tree sampling lab, we will do something similar to the quadrat or the transect option. We'll talk about that in a little bit more detail when we get to that particular lab. But again, it's a way to just sample a smaller area. And then for both of these, there are standard calculations that you would use to help you estimate how many might be in a larger area. If you have something that moves around, it might be really hard to find them, might be really hard to see them. Maybe they're nocturnal, maybe they like to hide. And so one way you can study a particular population or types of organisms is by setting traps. So what you would do here is trap your animal, use whatever method you are using to take your information about them and then release them back. So this is really the trap that you would use is really dependent on the type of animal. So sometimes you would use box traps, you might use net traps, you might do pitfall traps. And really what you're doing here is finding a trap that is going to be the most humane way to catch these animals in order to get their life history information, get the number of organisms. You can take an estimate. We are going to be using the mark recapture method, so this is one way to capture organisms. And really what you're doing here is trying to choose a humane way and a reasonable way to catch your animal. So these are going to depend on the type of animal that you have. You're not responsible for knowing specific trap types and animals. It's just kind of to let you know how ecologists are studying particular animals. So in our lab today, or this week, we are going to be using the mark recapture method. So this is a method where you tag an organism, somehow mark them so that they can be recognized. And you might tag these for some other reason besides mark recapture. Maybe you're just keeping track of the organisms in your population. Maybe it's a radio collar or something similar so that you can follow these organisms. But when we're using the mark recapture method, the goal is to tag them the first time you capture them and then go take a second sampling. And in the mark recapture method, you are estimating the population size by the number that you recapture in your particular area. So there's an example here on this mountain goat of an ear tag. And this works because basically you are assuming that once you release them, these animals disperse amongst their population. They spread back out to their natural area. And then you have an equation that you can use to help you estimate that population size based on the ratio between the unmarked and the recaptured animals. So the general idea for this method is to use an equation where you can calculate the population size based on these recaptured individuals. So for our lab, we are going to be using two different equations. We've got the Surber equation, and then we've got the Lincoln-Peterson, Peterson equation. One is a modification of the other. Lincoln-Peterson is actually this equation that we have here. That your sample size is going to be the number of marked individuals in your first catch times the second catch, and then divide that by the number of marked recaptures in the second catch. So for now, this is just to show you that we are illustrating the relationship between our numbers of caught organisms and the numbers that have been recaptured. Once we get formally into our lab, we will be looking at a slightly modified equation called the Surber equation, and it is just an adjustment of this particular equation to be a little bit more accurate.